Yeah. Okay, I guess we'll actually do this. Oh, you're recording. Perfect. Yeah, so I think we'll start. Um, Everyone, welcome Silas Busch to our Superlab meeting today. He is currently a PhD student in the lab of Christian Hansel at the University of Chicago, but he's about to finish. He's got his defense coming up very soon. And then he's going to actually do a postdoc at Rockefeller University. And he's here today to present his really exciting work that was published last year, I think, in Science on Purkinje cell morphology. So without further ado, take it away.
Thank you very much. I'm so happy to be here. Um, Let me position things right. Please interrupt while I'm going with any questions. I know the communication can be a little awkward through Zoom, but it actually helps if I get a little bit of feedback. So feel free to just shoot questions out if you have them.
Can you see us in the room? Can you see our
Yes.
if we raise our hand, you can. Okay.
Yes. You're a little small, so you might need to make an exaggerated motion. But your cerebellum will help coordinate that.
Yeah.
So speaking of the cerebellum, I'm going to talk today mostly at a cellular level about the cerebellum, which, as you all know, I'm sure is involved in coordinating motor activity, but also many non-motor features of our function and behavior.
Real quick, do you wanna maximize your slides at the moment?
Oh, yes. Thank you.
Oh, perfect.
My project is focusing on the cellular level, and in particular on the Purkinje cell, which is the principal neuron of the cerebellar cortex. It's involved in integrating all of the input to the cerebellum, and then it produces the sole output of the cerebellar cortex down to the cerebellar nuclei and then out to the rest of the brain. So my project the story really starts with this image, which you're probably very familiar with. This is Cahal's illustration of a human Purkinje cell. When I first started my graduate work, this image really captured my imagination, and I was quite interested in the staggering complexity of the dendritic arbor here. And I knew that to some extent this was a true representation of the human, as Cahal was known for being particularly detailed in his caricatures of neurons and other brain structures. But we also know that a lot of his illustrations were amalgamations of various cells he saw over time. So a lot of the question that I and my advisor had at the beginning of this project was how true to reality is this? Because this is quite different than a lot of what we work with. So we work in the mouse system, and so the cells that I would end up studying looked a lot more like this. And I think you can appreciate pretty immediately that this is quite substantially different in a number of ways. And the immediate question there was, does this difference in morphology mean anything? Does morphology influence the function of these cells? You might expect, given their structure, that this would be pretty well known at this point, but actually, a lot of what we know about the way that structure affects function in Purkinje cells is at the subcompartment level. So we know a lot about cable properties and excitability within small dendritic branches, but we don't really understand much about how the larger shape of the dendritic arbor could influence function. And asking that question is a little bit challenging the typical idea of how the cerebellum is structured anatomically. And so to appreciate that, let's talk a little bit about the cerebellar circuit, or the cerebellar cortical circuit. And so in particular, these are the players that we'll be talking about today. So there are Purkinje cells, which, as I mentioned, are the principal cell type of the cortex. And they integrate two major excitatory pathways, one from a very large number of parallel fibers, over 50,000 even in the mouse. And they combine that with the input from canonically a single climbing fiber input, which comes from the olivary nucleus of the brainstem. So you can and so the defined anatomy here is this large difference between a wide array of parallel fiber inputs and then a very, very specific one-to-one input relationship between the climbing fiber and the Purkinje cell. And this has defined our understanding of cerebellar function since the 60s, when Marr started to define a anatomically informed theory of cerebellar function. And part of why asking about morphology here is interesting is given that so the parallel fibers are numerous in their input, but they project orthogonally to the plane of the dendritic arbor, which is flat. So it's large, but it's flat, and the parallel fibers project orthogonal to that. And so you can imagine that changes in the width or the height of a Purkinje cell might alter how many or how diverse in representation their parallel fiber inputs are. So that's one possibility that we thought about. Another intriguing possibility is that the morphology might influence the way that the climbing fiber arranges itself on the dendrites. So in particular, we focused on the climbing fiber input, and that was because of what we knew about how the one-to-one relationship develops in the cerebellum. So early in development, an individual Purkinje cell in green here receives many 7, 13, 15 climbing fiber inputs on its nascent somatic structure. And as little growing dendrites start to form, the climbing fibers compete with each other for synaptic territory, and they compete to translocate to the primary dendrite that begins to emerge through development. And then eventually a single climbing fiber, it has been understood, outcompetes all of the rest of the climbing fibers and is permitted to translocate onto the primary dendrite. And the typical idea is that there's one core primary dendrite for the climbing fiber to do this. And so there's kind of a two bidirectional influence here of the climbing fiber development and the Purkinje cell development. And this created this notion that a Purkinje cell has one primary dendritic arbor. It might arborize complexly into these fine little dendrites, but it's really one core dendritic process. And then that is innervated by one climbing fiber. And that provides a very strong input, but a singular input. But when I started patching cells and filling them with dye, so this is in acute cerebellar slices, I would sometimes find cells that looked like this in the mouse. So you can appreciate this cell has two primary dendrites coming out of the soma. And so that kind of raised the alarm bells for us. What might happen here? Would that afford something different for the climbing fiber translocation process? So I kept patching more cells, and I would see ones that looked like this. This looks a bit more like the first mouse cell that I showed you. I found ones that looked like this, where there's a single trunk coming out of the dendrite, but it splits into very segregated compartments quite early. And in theory, those could provide different territories for multiple climbing fibers to translocate. And then, like I showed you, there were cells where the primary dendrite compartments emerged directly from the soma. And at least in the case of these latter two cases, it seems like you kind of get a two for the price of one. Two dendritic compartments that can process information for the price of only one soma and one output. So this led us to wonder, well, what can this two for the price of one buy us? And in particular, what can it do? What can it buy a human when your dendritic structure looks like this? And again, we still are not quite sure whether this is true to the actual structure of a human Purkinje cell. Not a lot of somewhat surprisingly, not a lot of work has been done in the human to elaborate what their dendritic structure looks like.
Silas, question there. Does that neuron have two principal?
Yeah, and you could even say this one has three.
Yeah, or three, yeah.
this small process here. And additionally, it has this very proximal bifurcation of one of the primary dendrites. So if we just focus on this little territory here, there's a lot of space for the competing climbing fibers to isolate and be happy. So that's quite exciting if this is a true representation of how a human cell looks. So eventually I got some human tissue and I immunolabeled it. I used a slightly different method than Cajal, but here's an example of a human Purkinje cell in all its glory. And you can see it indeed has these two at least this one in particular, has these two primary dendritic compartments, and those tend to split into large compartments fairly close to the soma. So maybe Cajal was actually quite accurate in this depiction. And I found a few other cells, and you can see there's quite a range of diverse structures here. And you've got cells with single trunks, but that split into a whole number of compartments right there, right next to the soma. And then you've got, whatever this is going on. So the human is really, really quite diverse in terms of Purkinje dendritic structure. And another thing you can appreciate is how the distance between these primary dendrite compartments is quite great. So not only might you be getting some interesting features within just the core region right around the soma, but this could also be producing quite unique physiological features just by virtue of how large and spread out the whole dendritic arbor is.
How many microns is it left to right, these cells? What's the scale bar here?
Oh, so I can show you in even more detail later, but in this case, these scale bars are 50 microns. So these cells are easily 500 microns wide, tend to be even more than that. They can get close to a millimeter wide.
Okay, so that's the reason why you're patching them. Your field of view of your camera is not, or your microscope is not big enough, so you're patching them out.
Yes.
together.
Yeah, that's why it's this
Okay.
tile scan.
Yeah.
Yeah. So we really wanted to study this. This really raised the alarm bells that there could be some quite interesting physiology here. So the first thing we had to do was try and get our hands around defining this dendritic structure if we wanted to study how it could alter the function. So what we did was we created these three categories for Purkinje cells, and I think we can all appreciate that this is really a spectrum of structure, but we thought that this was a fairly intuitive and potentially informative way to separate things. So we've got our normative cells, which were defined because this is more the classic idea of how a Purkinje cell looks. It's got one primary dendrite, and that arborizes into smaller branches and forms largely a single compartment. Whereas split cells have a single trunk coming from the soma, but they have this proximal bifurcation that effectively produces quite segregated compartments. And then there are what we call polydendritic cells, which have multiple dendrites emerging directly from the soma. So these are mouse cells. This morphological framework works in the mouse. It also translates to the human. So this is a nice tool to compare the two species. And so the next question, what we had to do was figure out what's the relative proportion of cells of each type. And so I ended up counting many cells, over 6,000 across both species. And what you can appreciate is that essentially all of the human Purkinje cells are split or polydendritic. So they have this kind of multi-compartment 2-for-1 kind of structure. Whereas in the mouse, it's around half of them that show that, and really only a minority that are showing the more extreme version, this polydendritic structure. So in counting these cells, the way that I did it was by taking whole slices from both the mouse and the human and immunolabeling the whole slice and then reconstructing it a slice map like this. So the advantage of this is not only does it give us the total cell counts and relative demographics, but it allows us to map where different cells are by their type. And so here, what you can appreciate, particularly in the human, is this increase as you go from anterior portions of the hemisphere, so lobule 3. This is all in the hemisphere of the human and the mouse. As you go from anterior lobules, like L3, all the way to more posterior regions, so like lobule 8, 9 even, you can see that there's this increase in the rate of polydendritic cells. And there's actually a similar pattern in the mouse, even though the absolute numbers of each type are different. So this was quite a fascinating anatomical finding, and the fact that you might get different demographics in different regions of the cerebellum, which we know from some of the work done by members present. We know that there are functional differences between different regions of the cerebellum. They participate in different tasks to different extents. So this observation of what we've been calling an anterior-posterior gradient suggested to us that there could be some kind of a physiological purpose to this. That it's not just random, it's not a bug, it might actually be a feature of the system.
Yeah, that's how lateral are these slices? How many centimeters are you out from the midline?
We are perhaps 2 to 3 centimeters out. So as approximate to the middle of the hemisphere as I could get. And I'm also trying to maintain, or I'm trying to capture parts of the tissue that are more complete. I'm not getting perfectly intact human tissue here, but
And in the mouse, you're also kind of like mid hemisphere, basically.
yeah.
Okay.
Yeah. Right. So we think that there could be some kind of a functional component here. And I already told you the story about climbing fiber translocation during early development. So we've reached the end of what we can do in the human to some extent. And because we don't really have access to physiological recordings in human tissue at the cellular level, where we can interrogate the circuit, unfortunately. But we do have the mouse as a powerful system. And as you see here, the mouse does have some representation of these different cell types. So we decided to try and pursue the question of is there a functional role here in the mouse? So the first experiment was still anatomical, but it gets us closer to the physiology. This is largely a needle in the haystack proof of principle approach. But what I did was I injected a low titer of a tracer into the inferior olive. And what that did is it labeled with this tracer the axons, which are the climbing fibers from the inferior olive, of a subset of climbing fibers. Okay, so now a subset of climbing fibers are labeled with this tracer. And then I paired that, once that is expressing in the animal, I would sack it and do my immunolabeling experiment for calbindin, which labels the Purkinje cells, and VGLUT2, which labels all climbing fiber terminals. And essentially the goal here was to see if I could find a Purkinje cell that has both tracer positive VGLUT2 puncta, so that would be climbing fiber terminals on the Purkinje cell from a tracer labeled climbing fiber, and tracer negative VGLUT2 puncta. So this method wouldn't tell us how many climbing fibers a cell would get, but if it does have both of these kinds of tracer positive and tracer negative VGLUT2 puncta, it would tell us there are at least 2 unique climbing fibers from 2 unique origin cells in the inferior olive. And so this is an example of a polydendritic cell here, where one branch has these tracer positive VGLUT2 puncta, and the other branch, the other primary dendritic compartment, has just VGLUT2 puncta without any tracer label present. So this is the proof of principle for us. This showed that first of all, true multi-innervation is possible. And so, and for those who don't work in cerebellum, you need to appreciate that this essentially defies that we've defied the textbook at this point. So the long-standing dogma in the field is that a single Purkinje cell in the adult animal, after this pruning process is done, receives only one climbing fiber input. So that, and there was some evidence, you can find hints of this in the literature, that there might be some multi-innervation in adults. But you really have to look for it, and it hadn't been reported. So this is the first real documentation that this phenomenon happens in the adult animal, and it conforms with our hypothesis about how the dendritic structure might influence that. Okay, so now we've got a climbing fiber, or a Purkinje cell that has multiple climbing fibers. Like I said, this experiment is really hard to scale up. It's kind of a needle in the haystack for various reasons. So we saw an approach where we could try and see the rate at which there's climbing fiber multi-innervation in a quantitative way. So
Can I
we
just ask Silas real quick, does this tell us anything about where those climbing fibers originate? So can you say anything about whether they might originate from a similar place in the inferior olive, if they're close to each other?
I don't know. The way that you would do that would be to do the heroic tracer reconstruction of single axons that Sujihara and others have done classically, but that still would only tell you about the labeled climbing fiber, and you wouldn't know the origin of the unlabeled one. So unfortunately, no. It is likely that they are originating from similar parts of the olive, because we know that they tend to that adjacent cells within the olive tend to project to similar areas within the cerebral cortex. But I'm also telling you a story that defies some of what we understand classically about human or about cerebellar anatomy. So I'm also a little hesitant to give you a strong claim that way either.
Yeah, okay.
Was it generally the case, or maybe you're going to get to this later, but was it generally the case or universally the case that when there were two, that they hit different branches?
So all but one study that I'm aware of where you can find hints and evidence that there might be multi-innervation, we're using this slice technique that I'm about to describe. And with this, you don't know anything about you don't visualize the climbing fibers. You only get their signal output on the Purkinje cell. So there was one study that did a similar kind of tracing method, and in that case, they could observe that the climbing fibers were innervating different territories on the dendritic arbor. So that also supported this this idea.
Thanks.
But all of this is in very small numbers of cells. So what we were aiming for was to scale this up and to try and look at, you know, what is how frequently does this happen? So what I would do, I would get an acute cerebellar slice, and I would perform whole cell patch clamp so I could record currents and hold the cell at various potentials. And then I would stimulate climbing fibers as they arise through as they ascend through the granule cell layer subadjacent to the Purkinje cell. Now in this preparation, I can't see the climbing fibers. I can just barely see the somas of the Purkinje cells I'm about to patch. So it's tricky because you need to put your electrode in several places subadjacent to your cell that you've patched and try stimulating at different intensities to see if you can catch a climbing fiber, essentially. So these are example traces of what an excitatory postsynaptic current looks like, recorded from the Purkinje cell. On the left is an example where no matter where I position my stimulating electrode, no matter how strong I turn my stimulating intensity, which ostensibly could get fibers that are more and more distant from the electrode, In this case, I can only get one. So these are multiple traces stacked on top of each other, and every single current is the same size. So it's the same climbing fiber providing this innervation. But in some cases, you would get a recording where you would turn up your stimulus intensity and you would go from seeing one current amplitude and that would be consistent. And then suddenly you'd start seeing another one. And that second current that's larger represents the summed current of multiple climbing fiber inputs that you've now started to stimulate because you turned your stimulus intensity up. And so this is actually a classical way to do this kind of study. It's just always been done in the developing animals, and it's a way of documenting how complete the process of pruning has become in animals. But what I found by doing all of these recordings exclusively in adults after the end of the pruning window is that approximately 15% of the cells in the adult received multiple climbing fibers that I could detect through this method. So while I'm doing these recordings, I'm also filling these cells with a dye, so I can use a confocal microscope that's built into my ePhys rig, so I can image their morphology. And then I can split out the cells by morphology, and you observe that virtually all of the cells that receive the multiple climbing fibers are indeed these split and polydendritic cells, these cells that have multiple primary dendrite compartments. And this is done with a sufficient sample size to really give us some representation of how frequent this is. And so among those cell types, it's around a quarter of cells that appear to get multiple climbing fiber input. We can further link this to the morphology by looking at more continuous variables instead of just the dendritic categories. We can look at the distance from the soma to the split. And what we see is that among cells that have multiple climbing fibers, that split tends to be much earlier. This also kind of works into the hypothesis that we had. If I measured the distance between the dendritic branches that form, the multiple compartments that form, you can see that the cells that have multiple climbing fibers tend to have wider, more separation between the compartments. And for the polydendritic cells, the angle between these multiple trunks emerging from the soma tends to be wider. So this gives us a sense that there really probably is a very important role for the morphology here. It's a very tight link between these fine morphological features and the prevalence of multi-innervation. So this is pretty critical for cerebellar physiology. The idea of how the climbing fiber works in the system is that this is supervised learning in action. So these parallel fibers are providing a wider contextual input to the Purkinje cell, and then the climbing fiber provides a supervisory signal, a teaching signal, you might say. And so when parallel fibers are active at the same time as a climbing fiber, the climbing fiber produces a very strong calcium influx into the dendrites. You can almost think of it like a backpropagation in pyramidal cells, except it has the inverse effect on plasticity at the parallel fiber synapse. What it does is it depresses them. And so the climbing fiber is providing this crucial signal to adjust the weights of the parallel fibers and alter the signaling properties of that Purkinje cell down to the nucleus and then out. And so what we're observing is that there might be Purkinje cells that are getting multiple teaching signals on their dendritic arbor. And so they could be selectively controlling different parallel fiber inputs instead of all the parallel fiber inputs on the tree being exposed to the same teaching signal. So to try and answer this, we wanted to look at how the signaling within the dendrite would happen in vivo. So to do this, what I did was I got a sparse. This was crucial, is that I got a sparse GCAMP label specifically within Purkinje cells. And the sparsity is important because I needed to be able to isolate individual Purkinje cells so I can know this two parts of the in the field of view are connected to the same neuron, right? And they're not part of different neurons. So that was a crucial component here. And so these are traces, examples from a polydendritic cell, the one depicted here, and then a normative cell, which I'm not picturing, but you can believe me. And so there are two traces for each of these, and the two traces represent two sides of the dendritic arbor, which I can reconstruct and show that they connect to the same cell. And so in green is one side or one branch, I'll call it, and in gray is the other branch, and below is a subtraction of the two from each other. And so this is the calcium signal, and the calcium signal is largely representative of the climbing fiber input. It's the climbing fiber that provides the really, really strong input that produces very strong calcium signals within these dendrites. There might be a little bit of influence from parallel fiber input in influencing the calcium signal, but we suspect that it's not quite as robust as the climbing fiber. But so what you can appreciate here is there are quite a few cases. Well, so typically when people are doing GCAM imaging in the cerebellum, they'll draw an ROI that captures the whole dendritic arbor. And so they're assuming that each Purkinje cell is going to look like this normative cell, where the two sides of the arbor are behaving identically. And so if you draw an ROI around the whole thing, you're just capturing the whole dendritic signal, and it's probably going to improve your signal to noise because you have a bigger ROI. Great. What I did was I was drawing these independent branch ROIs, and when I did that, I could observe that there were sometimes quite strong calcium signals in one branch that were not present in the other. And so our claim is that the only way that we could really be seeing this, and in a way that is specific to some cells and not others, is because this is the result of two different climbing fibers with non-overlapping input that are selectively activating different branches.
Silas, in this example, it looks like the two calcium signals are pretty correlated across the two branches.
Largely,
They
yes.
have very strong positive correlation. Now, I mean, do you have any intuition about how much that's your measurement process, or like incomplete separation of the branches, basically? Or to what degree is this? I mean, how would that look like if the two climbing fibers were firing independently? Right? Do you think you would get a zero correlation in the calcium signal if they really were totally independent?
Right. So I don't, because and this gets to the point that Caro raised earlier, which is that I suspect that the multiple climbing fibers are coming from similar areas of the olive, and really the olive is you've got a lot of gap junction coupling, you've got a lot of coupled activity within the olive. It's maybe less than people make it out to be, but it is still a robust phenomenon. So presuming as I do that these two climbing fibers are coming from similar areas of the olive, I wouldn't expect them to have completely uncorrelated activity. Regardless of the dendritic signal, I wouldn't expect their correlation to be zero anyway. So I expect there to be maybe 80% overlap in their signal up to that. And so that's what I observe in the dendritic signal, is that it largely overlaps, but there are quite a few instances, 10, 20, 30% of the time when they appear to be distinct.
Okay, so your average correlation between the two branches, if you can isolate them, is somewhere around 0.8 or something? Is that?
I could actually pull up.
Not that important, just like as a ballpark.
So one thing that I did was, instead of, well, so let me show you this figure, which is, this is just a characterization of how many events there were that were specific to one branch as a function of the total number of events. So this starts to get at that a little bit.
Yeah.
But a similar test that I did was doing exactly what you're suggesting, and just drawing the correlation between the signal and the two branches and looking at the strength of the correlation and stuff like that. And what I saw doing that, I don't remember the exact numbers, But what I saw when I did that was that the split in polydendritic cells, the correlation went way, way, way down, on average. And for some cells, it would really start to approach zero. I think and we could debate for a while how I should do that. Should I just be looking at the peak amplitudes and of the trace and correlating that, or should I really correlate every feature of the whole trace, just a Pearson correlation of the raw trace? We would expect there to be some difference in the noise. The ROIs are not always the same size and various other things like you can expect. So we start to lose a little bit of interpretability at that point. Yeah.
Why did you choose Cruise One? Is that just because that's the easiest to probe? Because they're doing a motor task, right? They're running while you're
Well,
recording.
so in this experiment that I'm showing right now, it's resting. They're resting.
Okay.
No. The treadmill is there to make them more comfortable. They can run if they want to, but I would collect my images when the animal was resting for this experiment.
Okay.
Yeah. So the reason why Cruise One, we know that there's a lot of, so this will give away a little bit of what my future experiment is going to be that I'll describe later, but we know that there's a lot of multisensory processing there. And so that would give us potentially a way to probe the different representation that the climbing fibers could provide. But so that is actually getting to the next experiment. So that's a great question and a great way for me to segue. But first I'll also say that the presence of local events appears to be additive. And this is another thing that you might predict if these are two independent climbing fibers. So climbing fibers tend to fire spontaneously during rest at about a 1 hertz frequency. It's about 1.2, but whatever. So what you might expect is if you're getting multiple climbing fiber inputs, unless there's something different in the olive, you should get a slightly elevated event rate, right? And that should correlate approximately with the lack of correlation between their two signals. So as they become less and less correlated, you're now getting multiple asynchronous 1 to 2 hertz signals. So when looking at the maximum event rate in a trace, I could see that when you have more local events, you would also just get a higher maximum event frequency. And so that suggests that this phenomenon is additive, which we would expect if we are adding climbing fibers, essentially. And this also somewhat suggests that maybe there's an aspect of increasing the dynamic range of climbing fiber output of complex spiking, which is the physiological output that the Purkinje cell produces when it's stimulated by a climbing fiber. So we found that quite interesting. Okay, so now to get to your question. We wanted to know what the possible purpose of this could be. Why have multiple climbing fibers that could differentially innervate different branches and provide these different calcium signals, for example. So what we wanted to do was test whether they are representing different receptive fields. And so the way to do that, this is another kind of proof of principle experiment. It's not ideal for several reasons, and you'll see why. But what we landed on was the whisker receptive field, which is highly reproducible across animals and across experimenters. And we know that climbing fibers tend to represent one or a small number of whiskers. So ostensibly, we wanted to see if we could stimulate individual climbing fibers by stimulating individual whiskers, and then potentially separate the signal of one climbing fiber from another in this way. So I don't know if you've ever tried to grab and stimulate a single whisker in a mouse before, but it's actually impossible. So what we did was we sedated the animals. So this is under ketamine anesthesia. So that's a lot of why this is not a perfect technique. It doesn't tell us accurately how real representation is working, but it allows us to survey the receptive field structure. So this is an anatomical experiment in a live animal. So with the animal sedated, I would stimulate 7, 8, 10, however many I could get while the animal was in a steady level of anesthesia. And I would stimulate them repeatedly because we know that under anesthesia, the representation is diminished. So the activity of the climbing fibers might be very reduced. So if I just stimulated them once or twice, the success rate would be extremely low. So what I did was a 2 Hertz stimulation for 50 seconds. And I would do that, and I would see an elevation in activity relative to 10-second baseline spontaneous period. And what I was looking for was, again, differential signaling across the dendrites. And so this is an example from a split Purkinje cell with two segregated branches, where you can see that this green branch appears to be responding quite frequently to the stimulation of this particular whisker, whereas the branch 2 does not really do that as much. And when we do this across many cells in many mice, we can see that we observe that kind of a local response much more in these split and poly cells. And you can probably see that this relative difference between the groups is actually looking quite a bit like the previous experiments with spontaneous activity. So this was a nice little proof of principle. And another thing, so when analyzing this, we were able to segregate responses as either global, so we can see that both dendrites are responding strongly to the stimulated whisker, or what we call a lateralized response, and that's like what's depicted here, where it's only really represented by one branch of the cell. And when we split that out, as you might expect from the previous graph, we see a lot more of these lateralized responses in the split and the polydendritic cells.
See, like when you have lateralized responses, that's kind of a single dissociation, right? You have one whisker that produces more response on one of the two branches. Do you ever have, I mean, did you find cells where you had, I mean, since you stimulated multiple whiskers, where you actually could show that there's a double dissociation, meaning like the other whisker would stimulate the other branch, and you could kind of do you not?
So I didn't have cases where there was a really so part of the issue with this experiment is how infrequent the response is, and so it really reduces my statistical power to determine, is this a response above what I would expect just during spontaneous activity. And so then when I'm asking the next level question that you are, and I did, it's a great question. I was demanding even more confidence that one branch was representing a particular whisker and the other was representing a different one and not the other one. So at that point, I just didn't have statistical power with the way I set up this experiment to say that clearly. Anecdotally, yes, I saw what looked like it could be that, but I couldn't prove that statistically here, unfortunately. But that would be a brilliant follow-up experiment. Yeah.
Yeah, because here it could be that one of the climbing fibers is just slightly less responsive to the stimulation. They both have the same kind of tuning functional responsiveness, or response properties, but one is just less responsive to your whatever you're doing out there, and so you get the lateralized response, right? So that
Right. Yeah, and that's possible. That's possible here. Yeah. But so one, and a lot of our problem here is that we're not looking at this in the awake animal, where we're getting a better representation of real signal processing. So the next experiment that we wanted to do, how am I doing on time? Looks like 45
We've got another 15 minutes.
Okay.
15 minutes.
Okay. So just briefly, cells that had more local events, and I would determine this during the spontaneous period. So this is like that first experiment with spontaneous activity. So cells that had more of that also appeared to be responsive to more whiskers. This starts to get a little bit at what you were asking, about whether it's just you have two climbing fibers and one is less responsive, but to the same whisker. So given this, we see that the cells that have more of this these local events and thus are more likely to be multi-innervated also tend to respond to more whiskers of the ones that I tried. So maybe they are actually surveying a larger number of whiskers overall. To say that really confidently, I would want more data, but this starts to push us in that direction of interpretation. Okay, so now we want to try and do an experiment in the awake animal. Whiskers are off the table now. We don't want to trim them because we're afraid that we might really change how the climbing fiber representation might work with trimmed whiskers. So we went instead for multisensory stimulation. And this gives us the even greater advantage of working in Cruce1 in the mouse. The whisker experiment was in medial Cruce1, which has more whisker responsiveness in the mouse. This is in lateral Cruce1. So I exposed the animals to all sorts of different combinations of multisensory stimuli while they were awake, and again resting. So I tried to isolate this from motor signals. And so what we can observe, so these traces are the two branch signal from this cell for different kinds of multisensory stimuli combinations. And the way I've broken it out is to show you what different kinds of responses could look like. So this is a global response where both branches were strongly responsive to this combination of all three sensory inputs. This one is the most robust case where a single branch responded to this light modality and the other does not. And then these are examples where one branch appears to be much more strongly representing this combination of sensory stimuli than the other. And this is this is not a binary phenomenon of, one climbing fiber represents one modality and this one represents another, but it's still quite surprising to see such a strong difference between the amplitude in the two different dendrites. And this could have differential ramifications for plasticity and other phenomena, other computation within those dendritic compartments. So like in the previous experiments, we saw more local events in split and poly cells. Here I've combined them into a single population. And interestingly, so uni here refers to just unimodal input, so like just a light or just a tone. Multi is combinations of them, and you actually see slightly more distinction between the two dendrites when you're working with multisensory stimuli, which is a little surprising to us, because you might predict that once you start combining stimuli, now you're going to be more likely to have similar representation across both dendrites because you're stimulating all the possible things that could excite the two climbing fibers. But possibly at this point, you're invoking other parts of the circuit that might help add branch-specific responsiveness. We just don't know at this point. So that's just an interesting tidbit. So that's in the awake animal. So now here we're showing that the awake animal can be differentially, to some extent, differentially encoding different sensory modalities across its branches. And do this in the awake situation. So let me summarize what I showed you. First, 15% of adult Purkinje cells in the mouse receive multiple climbing fibers, so it's not universally a one-to-one relationship. This lack of a one-to-one relationship is dependent on morphology. I showed you that the presence of multiple climbing fibers appears to be allowing these cells to multiplex distinct, at least to some extent, distinct teaching signals, which could differentially influence computation within the two dendrites. The morphological demographics are quite different in the human than in the mouse, and in particular, it's an exaggerated presence of these exact cells that appear to be particularly susceptible to getting multi-innervation and complex branch signaling. And then there are even more of those particular cell type in the posterior regions of the cerebellum in the human and in the mouse. So I can stop here and take questions. I've got a little bit of new data that I could take you through quickly if you're interested. It's a closer anatomical dissection of the human and the mouse in comparison.
I have some questions about, I
yeah.
just have some logical questions. I mean, I'm curious about, I'm not a cerebellar physiologist. I deal mostly in, well, basically in the cortex and the tactile periphery, but what was meant to be the function of having a single climbing fiber? Like what I'm trying to understand is the idea that you get multiple innervation is interesting. Ultimately, that needs to be communicated out through the Purkinje cell through the deep cerebellar nucleus. So like the multiplexing at the input is one thing. Whatever that means downstream, that thing can't tell which of the branches got affected. And I feel like in order to understand that, the simpler question is, what was it supposed to have meant that the Purkinje cell only had a single climbing fiber originally? What does the textbook say about the logic of that? And then what do you think this is changing about the logic of that?
Right. That's a great question. So a lot of the way that it was conceptualized in the literature is that the Purkinje cell is a bit like this perceptron structure where it's getting all of this parallel fiber input and the receptive fields so the parallel fiber field is supposed to represent a wide array of contextual information that could relate to different situations, right? Because it's representing a spectrum of information. And then you've got a selective teaching signal that is going to that it's for a given moment can update your contextual field and how much it's in, some parallel fiber inputs are influencing the Purkinje cell over others. So you have the flexibility and representation of Purkinje cell output come from the parallel fibers, and don't get me wrong, the parallel fibers influence the Purkinje cell output significantly. So Purkinje cells are spontaneously active. These are inhibitory neurons, and they're spontaneously active at 40, 50 hertz or so. And then bursts of parallel fiber input can increase the spontaneous rate, and then that is picked up by the cerebellar nucleus, where maybe 20 to 40 Purkinje cells converge on a single nucleus cell. So you get this diversity of representation that the parallel fibers can provide, and that's providing your distinct signaling. Complex computations that in the Purkinje cells can represent different phenomena. The climbing fiber is providing another way to think of it as an error signal. So specific to a certain situation, if you're trying to coordinate a movement, the parallel fibers are encoding different aspects of that movement property, the kinematics and stuff like that. And then the climbing fiber can selectively produce LTD at the parallel fiber synapses that are active and contributing to a mismatch in the motor function. And so that corrective signal is now diminishing the impact of those on the Purkinje cell, and then you can better coordinate the movement to be smooth in the subsequent based on the subsequent parallel fiber activity.
So if I'm hearing you right, that climbing fiber input induces plasticity in some subset of synapses that are active at some given moment in time.
Yeah, exactly. There's a narrow window in time
I see.
when coincident parallel fibers are influenced by and depressed
Gotcha.
Synapses are depressed.
Okay, so in that model, it's kind of intuitive that adding more is, I mean, I guess you lose something in there, but you have a ton of synapses, but now you have two training signals that give you kind of a richer, potentially richer capacity to train up some subset of those synapses. I mean, kind of, is that the right direction? Okay.
Well, that doesn't make sense, right? So,
Okay.
so you have two climbing fibers, and say like just so I'm thinking about the climbing fiber purely as a teaching signal now, right?
Okay.
So normally the one climbing fiber paradigm is that, well, this is the one loss function for the cell. It tries to predict its climbing fiber input. And ideally, it's going to lower its firing rate in anticipation of the climbing fiber coming in. And ideally, it's exactly the negative image of the climbing fiber. Now you have two climbing fibers. Let's assume they're uncorrelated. So one half of the dendritic branch tries to predict climbing fiber A, and the other branch climbing fiber B. Now, they only have one output, right? So they can never be as good as a climbing fiber that only has one Purkinje cell that only has one input. That could learn to predict the climbing fiber perfectly. This one has to compromise, right? Because it can't either predict AOP. So that's kind of weird. Maybe that's the reason that these two climbing fibers are usually in some overlapping agreement, where this might just be a trick to get more climbing fiber input into the cell. So that could be one thing. The other thing, I mean, Michael Heuser's lab, I can't remember the papers, but I mean, they've looked in how within the dendritic arbor things are integrated in the Purkinje cell, right? And I can't remember, I don't know if you thought about, so if you have two branches and they come together either in the split or polycell close to the cell body, how do they combine?
Mm-hmm.
And how does that, is this like an end gate? So you're gonna have to, like, both arbors have to agree that this is now a good place to send a simple spike, or you know, what's happening there? Right.
Yeah.
So there you could have actually, like, if you have nonlinearity there, then this could actually computationally give like new capacities, right? That you don't.
That's a fantastic point. So this also gets at a more contemporary understanding that the climbing fiber signal is not just an all or nothing binary, it's on or off. Right. So you can get different bursting behavior in the climbing fiber, and that's going to produce slightly different waveforms of the complex spike, which will so complex spikes in the Purkinje cell are a single really strong sodium-based spike, and then you have a series of little spikelets at the soma. And some of those spikelets are actually transferred down the axon as typical spikes. So you actually get a small burst of spikes every time you have a strong climbing fiber input. And then bursting of the climbing fiber can expand or diminish the size of that. So actually, the Purkinje cell does have a non-binary way to represent climbing, even a single climbing fiber bit. So that gives us a little bit of richness. Yeah, I love your point about, I mean, so essentially, these multi-dendritic cells are expressing a logic gate, potentially. We don't know. But this could give them that capacity to differentially represent one, the other, both. One climbing fiber tends to be stronger than the other, which could produce differential representation. Given the size of the currents that I recorded in the slice, and it's a little hard to say exactly given some details of the protocol, but the currents were generally large enough that I would expect even the weaker one to be able to produce a complex spike, but it might be diminished in size and how many spikelets it would produce and such like that. Of course, a lot of features of the complex spike are dependent on the axon, so it's not based on the climbing fiber per se. But so there could be some different representation there. Yeah. And so to get to another point that you raised, so one of the things that we're thinking about, what does this mean at the nucleus? So one theory that we have, and this relates to essentially, like you said, just having more climbing fiber representation, regardless of what exactly it's signaling. I think it's possible that this could be creating increased contrast within the nucleus. So Purkinje cells are inhibitory. And if you're getting more input to the Purkinje cells that converge on a nucleus cell, you are probably, I don't know for sure, but you're probably getting more asynchronous input across those converging Purkinje cells. So you're getting a more sustained period of asynchronous input, and Indira Raman's lab has shown that that's going to strongly inhibit your nucleus cell, and potentially more strongly because you have more asynchrony within your Purkinje cell population now. But then when you have a strong enough signal that everything needs to act in synchrony, and we know that there's a lot of also synchronous recruitment of Purkinje cells, suddenly you're going to get very strong signals within these Purkinje cells. Maybe both climbing fibers are recruited simultaneously. And now you get a strong, relatively brief converging Purkinje cell inhibition of the nucleus cell, which Raman has also shown produces this rebound excitation. So part of what I'm wondering, this is all speculation, but part of what I think this could be doing is essentially lowering the baseline nucleus cell activity to increase contrast during those periods of synchrony within the cerebellar cortex. But these are all fantastic questions to keep asking, and you can test this in animals. You can test this a bit in Slice. I'm hopeful that some people who do cellular and circuit level modeling can try and help us address the question. Yeah, there's a lot.
Do the climbing fibers branch? Do they themselves innervate multiple Purkinje cells?
Yes, and
Okay.
so mostly they branch early in the white matter, and then they'll innervate maybe 7 Purkinje cells, and generally in about the same area, although they can be pretty distant from each other.
So in the tactile periphery, we've studied these neurons that innervate Meissner corpuscles. They branch. Two interesting things happen. They branch extensively in the skin and innervate multiple mechanoreceptors, and it turns out multiple neurons innervate the same mechanoreceptor as well. And so a lot of these same issues about like how do you combine across, what's the computational advantage of having this branch? You lose, of course, individual cell specificity, but you might gain something. All the gains come out at the population level. So like, if you think about what's the advantages are like it's not what any individual single neuron can gets out of that. It loses spatial acuity, but coincidence across those becomes an extremely powerful way then to signal them. So yeah, I mean, it's kind of a very interesting, and the logic there is just that you have a limited space in the nerve, and so you're gonna have a situation where you can't pump as many neurons in the nerve as you can stick into the skin, and so there's a real computational advantage just by having more receptors. So let's say you have more receptors in the skin. Anyway, so I'm just curious about again, I know that there may not be an answer to that, but the level at which the population level signal and in the output nuclei matters, right? Because the single neuron thing is one question, and then how is it represented across the population is another.
Yeah, it's a fantastic question. We operate pretty much entirely at the cellular level. So I'm not expert at
Yeah, it's a career.
but now it can be done. Now that we have some detail about the cellular level.
Cool.
Great. Thank you so much. I think this was really fantastic. Thank you so much for talking with us, and we'll let you go. Safe travels wherever you're going now.
Thank you. Thank you for all your great questions and for having me. I'm really honored to be here with you. Thank you.
Thank you. This was great.
Thank you.
So Silas, you said, I mean, we can stop, but